Welcome to my talk on the impact of population density on sexual selection in the red flower beetle. I'm Lennart Winkler. I'm a PhD student in the Applied Zoology department of the TU Dresden. And I'm mainly interested in sexual selection and its role in adaptation. And we define sexual selection as selection arising of the competition for access to gametes or the resources provided by the partner. And so sexual selection led to the evolution of such extraordinary traits, for example, these amazing ornamentations, these strong sexual dimorphisms, these beetle penises with spikes all over them, and of course, male armamentation for male-male combat. But recent studies have also shown that there are quite a lot of differences in the strength and direction of sexual selection between populations and also between species. But we still have quite a limited understanding what causes these differences. And one of the main parameters that varies a lot between populations is demography. And that includes, for example, population size, sex ratio, dispersal, age structure, and density. And in our current project, we were mainly interested in the factor of density and its role in the strength of sexual selection. And while we know quite a lot about the effects of, for example, sperm competition or sex ratio on the strength of sexual selection, we know surprisingly little about the influence of density, although it varies quite a lot between populations. But of course, density is quite closely connected to population size, because if you imagine you increase the population size but keep the habitat area constant, then you of course also increase density. And both of these factors might influence mating behavior, the strength and direction of sexual selection, and ultimately explain intra and interspecific differences in sexual selection. So our question was, does density influence the strength of sexual selection in the red flower beetle? But how do we actually measure sexual selection? So we used three main metrics, standardized metrics, so they are quite useful for comparisons between studies and between organisms. And we used the opportunity for sexual selection, the Bateman gradient, and the Jones index. And here you can see some example data. In blue, you can see data for males. In reddish for females, you can see the mating success here on the x-axis, and on the y-axis reproductive success. And here you can see the variance in mating success for males and females, and that's known as the opportunity for sexual selection. So the opportunity for sexual selection is defined as the variance in relativized mating success, and it sets an upper boundary for the strength of precopulatory sexual selection. And if we look at the relationship between reproductive success and mating success, we get the Bateman gradient. So that is the relationship between standardized mating success and standardized reproductive success. And it measures basically the fitness benefit of multiple mating. So it measures with each additional mating how much more fitness does an individual get. And what Bateman also in his original study on this topic formulated is that we would expect typically the Bateman gradient and also the opportunity for sexual selection, so the variance in mating success, to be larger in males compared to females. Because in the example of the Bateman gradient, for males, they should often be mainly limited by their access to gametes in their fitness. So an additional mating brings quite a lot of fitness return for males, while an additional mating partner for females often does not, because in many systems, female reproductive output is mainly limited by their resources and not by the number of mating partners. So as we see in this example, we typically expect a steeper Bateman gradient in males compared to females. And finally, we use the Jones index, and this metric has been shown to be quite a good indicator of the strength of sexual selection. It measures the maximum strength of sexual selection. It's basically a combination of the Bateman gradient and the opportunity for sexual selection. So what did we actually do? We manipulated area and population size in a full factorial design, and we kept the sex ratio equal throughout our experiments. We had one focal per group and some reindeer competitors. The reindeer means basically it's a genetic marker that we can use in Tribolium castaneum, the red flower beetle, our model system, to assign parentage to the focal or the competitor. Now we had some wild type mating partners, of course, too. And we recorded the mating behavior and counted the offspring, so we get data on their mating success and their fitness. And these are the 4 treatments that we had. On this axis, we have a decrease in area, so we decrease the mating arena size. And on this axis here, we have an increase in the population size, so the group size. We have 6 beetles and 12 beetles for the large groups. So we had 4 different densities that we can compare. We manipulated area and group size in a full factorial design. So let's look at some results. First, the opportunity for sexual selection. I will always first talk about our results on group size, and then secondly, talk about our results on the habitat area manipulation. So first, group size, the opportunity for sexual selection. Here on the y-axis, you could see the opportunity for sexual selection, and that is the variance in relativized mating success, setting an upper boundary for the strength of precopulatory sexual selection. And you can see the data for focal males and focal females, and you can see that in both we have a significant increase with increasing group size of the opportunity for sexual selection. In females, this effect is even stronger than in males. So we see an increase with increasing area in the opportunity for sexual selection. And then if we secondly look at our area treatment, so where we manipulated the area of the mating arenas, we see that there was no significant effect of our treatment in females or males. So in summary, we find that group size, with increasing group size, and by that increasing density, we find an increase in the opportunity for sexual selection. But if we increase density just by manipulating the habitat area, so in this case, the mating arena size, we find no difference in both sexes in the opportunity for sexual selection. Secondly, we looked at the Bateman gradients. So here you can see on the x-axis. So first, again, the results on group size, and then on the next slide, we see the results on area size manipulation. And on the x-axis here, you can see relativized mating success. On the y-axis, reproductive success. And the relationship between these two is the Bateman gradient, measuring the fitness return of additional matings. And you can see in this small groups here on the left, we have a significant sex difference with a steeper Bateman gradient in males compared to females. And that is what we would typically expect from sexual selection theory, that males benefit more from an additional mating compared to females. But if we look at the large groups, we see that in red, the females compared to the blue line here for males, the females have a steeper Bateman gradient than males, but not significantly so. And if we compare now our treatments, we find that for females, we find no difference in the steepness of the Bateman gradient between small and large groups, while for males, we find a significant shallower Bateman gradient with larger groups. So if we increase density by increasing group size, we find a shallower Bateman gradient in males. Secondly, we can also look at our manipulation of habitat area, and we find that in the large habitats, we see that females have a significantly steeper Bateman gradient than males. So that's quite surprising and unusual. And apparently in Tribolium castaneum, females can also benefit a lot by multiple mating. And what you see here is that in the smaller area, we have again a significant sex difference, but now with the increased density, we find that males have a significantly steeper Bateman gradient compared to females. If we compare between the treatments again, we find again that for males, we have a significant difference, but not for females. And this means that for males, we find a significant increase in the steepness of the Bateman gradient if we increase density by decreasing the mating arena size. So the opposite effect to what we've seen before. So in group size, we have a decrease in the Bateman gradient in males, while if we manipulate habitat area, we have an increase. So that's quite an interesting pattern here. Finally, we looked at the Jones index, measuring the maximum strength of sexual selection. It has been shown to be a good measure of the strength of sexual selection. And we see that here on the y-axis is the Jones index, and we have again males and females. Again, first looking at group size, you see that in females we have a significant increase of the Jones index, so stronger sexual selection with increasing density by group size, while we don't see this effect in males. And if we look at our area treatment, again, we see that for females now we don't find an effect. So an increase in density by decreasing the mating size, the size of the mating arenas, we don't find an increase in sexual selection in females, but we find a significant increase in males with stronger sexual selection in the smaller arenas, so with increased density. To summarize that, we find that density increased the opportunity for sexual selection with larger groups. We find that the Bateman gradient was affected only in males, but in different directions by group size and area. We find a significantly positive Bateman gradient for males and females. So also females benefit from additional matings in this system in Tribolium castaneum. And finally, we find that the Jones index, it was influenced by the density in both treatments, but for males and females differently. So the key messages of my talk are first that the density can influence sexual selection. So it seems to be an important parameter for the strength of sexual selection. Secondly, we find that the effect of density on sexual selection can be sex specific. And finally, third, we find that group size and area can influence sexual selection differently. So whenever we manipulate density by either of these factors, we have to take that into account. And group size tended to have a larger impact overall than habitat area. But we still don't really understand the underlying mechanisms of these, so stay tuned. We have data, for example, on the mating behavior, and also we did a variance decomposition to understand the different sources that cause the different selection pressures in the different treatments. So with that, I want to thank my co-authors on this study. I want to thank my PhD supervisor from the CNRS, Tim Janicke. I want to thank Ronja Alhard, my bachelor student at the TU Dresden. I want to thank my group at the TU Dresden and the DFG for funding us, and you for your kind attention. Thank you very much.